Next up, Part B asks you to predict the amino acid sequence of the protein made from this mRNA sequence. In this part of the problem, we will review translation. We'll walk step by step through an example and then the practice problem, showing how we take the mRNA transcribed from DNA and then translate that mRNA into the amino acid language of proteins. The first question we ask when looking at an RNA sequence we need to translate is, where do we start translating this mRNA? Where do we start translating the mRNA, you say? Where do we start translating, you say? Well, yes, that will be at the start codon. The start codon is the RNA triplet AUG. You'll always need an AUG to signal where to start translation. Translation will always start with an AUG start codon, and the first amino acid translated will thus always be a methionine, as you can see in this genetic code table here. So let's go back to our example and look at our mRNA. When we translate our mRNA into protein, we'll be reading it from 5' to 3', and unless otherwise directed by some specific instructions in the problem, we're going to start translating at the first AUG start codon we encounter. In this example sequence, that's here. We'll begin translating at the start codon, and we'll build our growing polypeptide from the N to the C terminal direction. The start codon, again, to emphasize, will translate to a methionine. So the first amino acid in our example protein sequence will be methionine. Then we read down the mRNA in the 5' to 3' direction and look at the next codon, that is the next triplet of bases, CCU. You'll use the genetic code codon table to look up which amino acid the CCU codon specifies. First, find the row for the first letter, C, then the column for the second letter, also C. This lets you zero in on the third letter, U. So the CCU codon codes for the amino acid proline. Proline will be the next amino acid translated into our example polypeptide. To finish translating, you'll continue this process until you hit a stop codon. So reading 5' to 3' on the mRNA to the next codon, we see it's CAU. To find the next amino acid to be translated, we go back to the codon table. The first letter is C, the second letter of the codon is A. Find where the row and the column overlap, and you can find the full codon CAU. Here we see it codes for the amino acid histidine. So histidine will be the next amino acid in the protein sequence. Then you can go to the next codon on the mRNA. It's UAA here. Proceed to the codon table and find UAA. As you'll see when you look at UAA here, UAA is actually one of the 3 stop codons. Recall from lecture that stop codons do not code for amino acids. Instead, they signal binding of a release factor that causes the termination of translation. So our UAA stop codon here won't code for an amino acid. Instead, it means that our protein sequence ends as methionine, proline, histidine, and no more amino acids are added. With that review under our belt, let's then proceed back over to Part B of the practice problem and try the same process. Here's the mRNA we generated in Part A of the problem. To translate this mRNA to protein, again, the first step is to locate the start codon, which is AUG. The start codon will always code for methionine as the first amino acid in the growing polypeptide. Then just keep reading down the mRNA in the 5' to 3' direction, just like the ribosome does. The next codon or triplet of bases is GGC. If we go to the codon table, we can zero in on GGC. Find the row of the first letter, G, the column of the second letter, also G, and find GGC. And we see that this codon codes for the amino acid glycine. So glycine must be the next amino acid. Then look at the next codon, it's UCG. Flip back to your codon table and find UCG in the same way. UCG codes for serine. That makes serine the next amino acid. Shift over 3 nucleotides again, and your next codon in line is GCA. Find GCA in the codon table the exact same way. GCA codes for the amino acid alanine, so the next amino acid in the polypeptide will be alanine, and so on and so forth. The next codon, without working through the full process again, codes for the amino acid cysteine. The codon after that, however, is UAA. UAA is one of the 3 stop codons that signal termination of translation. There is no tRNA that can recognize the stop codon, so no amino acid will be added for the stop codon. Instead, stop codons in the mRNA will lead to the binding of the release factor, which will trigger the release of the protein chain and the end of translation of the protein. So that means that with this stop codon, we're finished, and we have completed the peptide. So this is the answer for part B of the practice problem. Now, let's proceed to the last part of the problem, part C. For the answer of part C, it's clear that they want a 3-nucleotide sequence of a tRNA anticodon that will pair with some mRNA codon. But there are key details about which particular tRNA anticodon they want. We need to find the tRNA anticodon that will be in the A site when the P site has another tRNA in it with a dipeptide attached to it. But what are the A and P site, and how do they relate to tRNAs and translating a messenger RNA into a protein? Let's quickly review the lecture material. The A site, P site, and E site are all sites on a large subunit of the ribosome. The ribosome is the complex molecular machine made of ribosomal RNAs and proteins that reads your messenger RNAs and translates them into newly formed polypeptides or proteins. What happens in the different sites? The P site in the ribosome holds a tRNA with the growing polypeptide strand covalently bound to it. The next tRNA comes in at the A site. This tRNA will have an anticodon that runs 3' to 5' and complementarily base pairs with the next mRNA 5' to 3' codon. The tRNA in the A site will also be charged or covalently bound to the correct amino acid that codon calls for. The ribosome will catalyze the cleavage of the growing polypeptide strand off of the tRNA in the P site and onto the amino acid on the tRNA in the A site. The polypeptide will have grown larger by 1 more residue, and then the ribosome reads 1 more codon down the mRNA 5' to 3'. The old tRNA, which lost the growing chain, moves to the E site where it can exit the ribosome and go off to be charged with another amino acid again. And now the newer tRNA, with the growing chain bound to it, is shifted to the P site, with the A site open again to receive the next tRNA with its amino acid, ready to repeat the cycle again. So with that refresher in mind, let's think again about what part C of this practice problem wants. You need to identify what tRNA will be the new tRNA that just came into the A site of the ribosome. And you want to know what that tRNA is at the point when the tRNA in the P site, that is, the tRNA with the growing polypeptide chain on it, has a dipeptide on it. A dipeptide is a polypeptide that has 2 amino acids incorporated into it so far. So let's modify the diagram from lecture to look at this. We want to consider the case where the ribosome has a tRNA in the P site with the first 2 amino acids of our protein from part B already attached to it. In other words, methionine and glycine. At that point, with the methionine and glycine dipeptide in the P site, we want to know what the tRNA newly arrived in the A site will be. Well, it must carry the next amino acid in line from the protein in part B, so it must be the tRNA for serine. But then, how do we figure out the 3' to 5' anticodon of this tRNA? Well, remember from lecture that a 3' to 5' tRNA anticodon is going to complementarily base pair with each 5' to 3' codon in the mRNA coding for an amino acid. So to find out the anticodon of the tRNA for serine in our A site here, we just need to do some base pairing with the mRNA codon that codes for that serine, UCG. The first U pairs with an A. So start the tRNA anticodon, which again will run 3' to 5' with an A. Then C pairs with G, and the G pairs with a C. Then there you have it. Your tRNA anticodon for the tRNA in the A site when a dipeptide is on the P site tRNA is AGC, running in the 3' to 5' direction. or CGA written in the other direction, 5' to 3' from the answer key. With that answer, we conclude this tutorial for BIOS E1A. Remember, if you have questions about this or any other practice problems for the course, you can post them to the course discussion forums online. And as always, thank you very much for listening.